Okay, so today we're talking about meiosis. So I'm going to try to keep this short but sweet, but still give you all the information that you need to know. So every time we talk about anything in meiosis, I want you to be trying to keep it separate, compare and contrast from mitosis. So meiosis is a special type of cell division. Doesn't happen in every cell. It's not a normal part of the cell life cycle. It's its own special thing, own special type of cell division. And it specifically happens prior to sexual reproduction. So its goal is to produce genetically different cells that we're going to call gametes, which can then combine during sexual reproduction to form a new organism that has totally different genetics from either of its parents. Okay. So very brief overview of sexual reproduction. So most plants and animals have two sets of chromosomes, one set from each parent. And we've been talking about them. We've been talking about them as our diploid cells with our homologous pairs of chromosomes, right? So I like to draw, even though I'm not good at it, right? So we have our homologous pairs, right? So whenever we see like this, right, that's a diploid organism. We know we have a version 1, right? So like 1A and 1B, right? So that's what it means to be diploid. And you're going to inherit one of each of these chromosomes from your parents. So you might have 1A from your mom, 1B from your dad. Okay. During fertilization, though, we want to be able to combine genetic information from each parent, but we never want to end up with an organism where we have like 4 of this same chromosome, right? Basically, we don't want to end up with genetic doubling and that information that would have to be contained in the nucleus growing exponentially because we're doubling every generation. That just wouldn't work biologically, wouldn't produce fertile offspring. It would mess with a lot of the cellular processes. So cells had to come up with a way to produce a diploid offspring, right? So this is where meiosis comes in. So meiosis is going to create these special gametes, right? These sex cells that are haploid. So they're going to be haploid. They're going to have only half of this information, right? And we're actually going to see, so I've drawn you a chromosome with sister chromatids in it. But when we see in our gamete, we might have 1A just being like that, right? We're going to have haploid gametes that have just one copy, so that then when they combine with another gamete from another parent, right? Say we got a 1B over here, we're back to a diploid organism. So this is helpful for maintaining the number of chromosomes from generation to generation. So you don't start out with 2 chromosomes and then get 4 and then get 8 and so on. So we need to have our information during this meiosis process. So haploid versus diploid. Here we can see a comparison in kind of cartoon drawings, right? Better drawings than mine. So we can see here this haploid gamete has 3 chromosomes here. This diploid somatic cell has 6, right? So if we wanted to circle them, right, like maybe this is a pair, right? So that could be chromosome 1. This could be chromosome 2. This could be chromosome 3, right? And it's a diploid cell. So it has 2 versions of each, a red and a blue. And our haploid gamete, we still have chromosomes 1, 2, and 3. But now we just have 1 copy. We're haploid. We don't have 2 different copies with different alleles. We just have 1 chromosome 1, 1 chromosome 2, and 1 chromosome 3. Now, note that this haploid gamete, it doesn't have only the blue versions or only the red versions. Which one of this pair ends up in each gamete is a random process. So that's part of what this is illustrating by showing an assortment of colors. And as you run through the meiosis problem set today, you'll be trying a couple different ways to divide things and recombine things, showing you how you don't always get the same result in every gamete. So that's an important part of how genetic variation gets introduced, is that it's kind of a grab bag of which version you get in a haploid gamete. Okay. But haploid gamete, half the stuff of our diploid somatic cell. Remember, somatic cells are normal body cells, right? A heart cell, a lung cell, an eye cell, the tip of your pinky cell. Gametes are just sex cells, so cells that are going to make sperm and egg cells. So most cells in the body are not somatic. Please do not tell me that a cell that you're describing to me is going to go through interphase and then mitosis and then meiosis and back to interphase. It doesn't work like that. That is never going to happen. So meiosis happens only when we're producing gametes for sexual reproduction from germline cells. Okay. So germline cells, right, start out as diploid cells, right? So we have these germ cells, they're diploid cells, but they're specifically for producing our sex cells for sexual reproduction, right? So this germ cell starts out as diploid. Through the process of meiosis, we have 2 cycles of division producing a haploid gamete. Right? And haploid and diploid are often expressed 2N versus 1N, saying like we have 2 versions of each chromosome versus we have 1 version of each chromosome. So our gamete, we have 1 version. That is the end of this process for this initial organism. What happens next to that gamete is not interphase, but sexual reproduction. So it joins up with another gamete. So from another parent, we call this process fertilization. And that produces a zygote and goes through all sorts of development, right? It becomes an embryo, becomes a fetus, becomes a baby, basically. So that's what's going on. And then all the somatic cells in that new organism undergo mitosis. So that's how we could relate all these terms together in a drawing if we wanted to, but it wouldn't be a drawing of your typical cell cycle. It would be a life cycle of a whole organism, not just one cell. Okay, so the key thing to remember for meiosis all the time, gametes are going to be haploid, and every gamete is going to be non-identical to the parent, right? So we start with a diploid germ cell, and we end up with a haploid gamete. So already, the cell we started with is not identical to the cell we end up with. So this is one of the big differences between mitosis and meiosis, is that we're producing non-identical gametes. We'll see they're actually not going to be identical to each other either. Okay. So in meiosis, our cells are going to divide twice. So all the chromosomes are going to duplicate the way they did before mitosis. So there's still an S phase before meiosis. So that's the same as it was for mitosis. What's going to happen during meiosis 1, so during our first cycle division, is that our homologous chromosomes are going to line up at the metaphase plate. So what we mean by that, and we'll see other pictures, right, is that instead of, like in mitosis, we had our chromosomes lining up 1 by 1 down the center of the cell, right? And we knew that we were going to pull them apart, right? Like that, right? We're going to have half go that way, half go that way. Instead of lining up 1 by 1, like they did in mitosis, we're going to have them line up 2 by 2. So the homologous chromosomes are lining up across from each other. And we might have blues on the top and reds on the bottom, or sometimes they might be flipped, right? Maybe our blue is on the bottom, maybe our red is on the top. The colors don't matter in that way, right? Okay. So then when we divide the cell, when we pull things apart, what's going to happen, right? We're going to have this chunk, of the cell go that way and become one of our meiosis 1 daughter cells. And this top chunk on the other side of the line is going the other way. Okay, so the homologous chromosomes are what are separating during meiosis 1. Now this is really important because this is what makes our cells haploid, right? The definition of a diploid cell is that it contains homologous chromosomes. We pull apart the homologous chromosomes during meiosis 1. As soon as we do that, we're going to end up with haploid cells. So these cells that are entering meiosis 2, even though they have sister chromatids, right, they still have this X-like structure, they are no longer diploid because their pair is gone, right? During meiosis 2, physically, if you looked at the cell, it would look a lot like mitosis because you would see a single file line down the center of the cell during metaphase of meiosis 2. So that would look like mitosis, but the reason it's not the same is that you have fewer chromosomes overall because you already split up the homologous pairs during meiosis 1. Okay. So we are going to look at each of the phases drawn out, but this is like a summary of the really important bits, basically. Okay, so here we see meiosis 1. So we see we're starting out in our prophase, which for our purposes, we're lumping with prometaphase as just one kind of big phase. We see we have our homologous chromosomes. So every chromosome has a red version and a blue version in this image, right? Just like we have red versions and yellow versions in our problem set. Okay, so during this first part of meiosis, we have our centrosomes spreading to the poles. We have our spindle forming. We'd now call this a meiotic spindle because it's for meiosis. And those microtubules, instead of catching individual chromosomes at the center, they're now catching on to these homologous chromosomes that have paired up. So we'll see those referred to as bivalents or tetrads sometimes. That refers to the fact that we've kind of stuck our two homologous chromosomes together so that during metaphase 1, when they line up on the metaphase plate, our homologous chromosomes are 2 by 2 instead of 1 by 1. Right? And note that these phases for meiosis, right, they have the same names as they did in mitosis, but now they have numbers in Roman numerals after them. So during meiosis 1, we have a 1, right? We have prophase 1, we have metaphase 1. During meiosis 2, that'll be a 2. So if you refer to metaphase 1 out of context, don't mention whether it's mitosis or meiosis, you'd still know because that's the name of this specific phase during meiosis 1. Okay, so they line up 2 by 2 at the metaphase plate, and then the cell is pulling apart into 2 cells, so it's dividing right along that dotted line. So during anaphase 1, we can see these homologous chromosomes separating. And then in telophase 1, we can see that this first set of daughter cells at the end of meiosis 1 now has fewer chromosomes in each cell than our original cell did, right? So this first cell was a diploid cell. Diploid, and I'll just write dip, okay? But these cells are now haploid, right? So we can tell because they have fewer chromosomes, right? So if we were to try to circle and count them, right, we'd see there are 1, 2, 3, 4, 5, 6 chromosomes here in this diploid cell at the beginning of meiosis 1. But here at telophase 1, when we have these cells separating, you can see that each of them is just going to have 3. 1, 2, 3. Right? So half of the 6 that we started with, right? So that's why this is 2n, this is 1n, and our n in this case would be 3, right? So 2 times 3 is 6, 1 times 3 is 3. That's what that notation is about, basically. Okay. Ooh. So that brings us to meiosis 2. So what we see here is that the daughter cells at the end of meiosis 1 are the cells we start out with for the beginning of meiosis 2. So that's why we see a double line of cells here. This is to emphasize, right, that each of these cells from the end of meiosis 1 is undergoing this process. Okay, so each of those cells with their 3 chromosomes, right, is then having those chromosomes attach to our meiotic spindle, right? So we again have that nucleus disappearing. We have the centrosomes moving to the ends of the poles of now our daughter cells during prophase 1, or sorry, prophase 2. During metaphase 2, we have our chromosomes lining up down the center, but there are fewer of them than there would be if this were mitosis. There are 3. In mitosis, there would be 6. So meiosis has half the number, right? So then in anaphase 2, it looks just like anaphase and mitosis did as well. We have our chromatids getting pulled to opposite ends, right? So we basically just cut this cell down the dotted line. And each of these daughter cells from the end of meiosis 1, now at the end of meiosis 2, forms 2 daughter cells, right? Which means we have a total of 4. It's actually kind of cool as you start to think about heredity, which we're going to talk about next time. You can kind of think about this, right? If you've seen a Punnett square before, right, in high school, so you actually basically have created a Punnett square with our 4 cells, our 4 daughter cells at the end of meiosis 2. Right, so that's a mathematically convenient when we talk about heredity, but it also says something real about the biology as well. So these cells are haploid. These are haploid gametes. These are the sperm and egg cells. Okay. This is a quick note about some things that can go wrong in these processes. So understanding mitosis and meiosis is important partially so that we can understand some genetic anomalies that occur and can cause big problems in people's lives, although some of them are definitely survivable. So when we say non-disjunction happens, what non-disjunction basically means is that the chromosomes don't separate right during either anaphase 1, or anaphase 2, right? So either during mitosis, during meiosis 1, or meiosis 2. And so we can see that if that happens, if we don't have the normal separation in meiosis 1 of our homologous pair, right, we end up with a cell that doesn't have any copies of that chromosome at all, 0 versions, and we have a cell that has double, right? Or if this were to happen during meiosis 2, we'd end up with something similar, but with these individual chromosomes, these chromatids. In mitosis, this would be less of a problem because it would only happen in, a single cell that's replicating, not a germline cell, not a cell that's going to be a whole organism. But we'd end up with similar problems where we'd have cells that just don't have the typical number of chromosomes. Right? So that's what aneuploidy means, is too many or too few copies of a specific chromosome, or polyploidy would also refer to a similar kind of process. So here are, in case you're interested, some of the syndromes related to cases of aneuploidy that are survivable. So some of them are more or less observable in people. Some of them are toxic. Some of them are embryonically lethal. But some of them are, probably not pleasant, but they're fine, right? Okay. Other chromosomal abnormalities that can happen include changes to the physical shape of the chromosome. Like sometimes during this process, we'll end up accidentally making an extra copy of some segment of a chromosome. So we call that duplication. So you could identify duplication here because this chromosome got longer, right? It has this segment repeated. Maybe something that shows up more, right? So this segment we can see has been repeated and this chromosome got longer. Can have inversion, which is a little trickier to see here, but chromosomes have specific banding patterns if you stain them. So you can see what happened here is basically this leg of the chromosome got ripped off, and then the cell was like, oh, that's okay, I'll tack it back on, but it did it backwards. So that's an inversion. A deletion would be if we took this segment, it got ripped off and we never tacked it back on. So it's just completely deleted. So we end up with a much shorter chromosome. And we can have all sorts of basically rearrangements of the chromosomes, right? An insertion would be if we have a chunk from the wrong chromosome get inserted in the middle of our chromosome. And translocation is kind of neat. We can have translocation happen when stuff jumps around within some chromosomes. So these are all various things that are kind of happening as we have this dynamic process going on as we're moving our genetic components apart, right?